share my screen. All right. Can you all see my screen?
Yes, sir.
Yes, sir.
Hello.
Yes, sir. We can see.
Can you all see my? Yeah. Yeah. Okay. Great. So yeah. So in the last class we were talking about detergents, and just to we quickly ran through this, but just for brief recap. So detergents are primarily molecules which are extremely ionic in nature. I mean, ionic meaning that their head group is pretty ionic, and they are yeah, and their tail is as usual pretty hydrophobic. And for example, here you have DPC is called as dodecyl phosphocholine. You have a phosphate group attached to a choline moiety, and this is charged. And yeah, so these detergents are primarily since they are since we talked about the correlation between the cross section of the head group and the tail group, and that determines whether these kind of molecules whether they form lipid bilayers or micelles. So these detergents, because of a polar head group which is very polar, pretty ionic, and a single-tailed backbone, I mean hydrocarbon chain. So these guys form micelles. And here we spoke about this sodium dodecyl sulfate. It's SDS, which is extremely useful. As you'll go, I'm not sure whether you'll be doing protein gels. Probably you might be this year. We don't know. So you will see that SDS is extremely useful in denaturing protein. Because as we were talking briefly in the last class, that this hydrophobic part, this hydrophobic tail binds to the hydrophobic core, or even not even core, at least initially the hydrophobic patches on the surface of proteins. And since this head group binds very strongly to water molecules, it sort of opens it up, and or destabilizes at least the core, at least the surface, initially the surface, and eventually you have some more space. These detergents can go in, bind to the core, and eventually completely expose the core and denature the protein. So SDS is always used when you are to denature protein, run protein gels, and analyze what sort of proteins you have in a complex mixture or even in a cellular lysate. Okay, and you have milder detergents, of which the head groups are not very ionic. You can see here dodecyl maltoside, where you have the maltose group here. It's not which is polar. You have the hydroxy groups, which can interact with the water molecule. Uh, Ionic, and likewise you have Triton X, and these guys are the ones which are very mild detergent, and that is why they are used to, it's not typically called that it's for denaturing protein, but rather it's for solubilizing membrane proteins, meaning that there are certain proteins which are embedded in the membrane, and if you are to study their structure, you have to sort of get them out of the membrane, in simple terms. And the way you do is you use these detergents in your solution, and these proteins can essentially transfer from that lipid bilayer on the surface of cells onto your specific vesicles or micelles or whatever, and you can take the protein out of the membrane and study their structure, function, whatever. Okay, so yeah, so this is how the dirt basically works. You would have seen, all of you probably would have seen advertisements in the television, where it shows that how essentially micelles, ionic micelles or detergents, take out dirt from your clothes, right? Yeah. So the other thing that so in the last class you might remember that I was showing you that how dynamic it was. Again, a molecular dynamic simulation, which sort of shows that these micelles, when you draw them, it looks like rigid, but you have to imagine that these molecules are not very rigid. They are quite flexible molecules. So likewise, if you are to consider lipid bilayers, now lipid bilayers are also sort of very flexible. Okay, Now, there are experiments. We'll talk about one simple experiment that demonstrates that, so there is a sort of diffusion across the membrane. So these lipids are free to move. Well, yeah. So yeah, we'll see we'll see in one of these experiments that clearly demonstrates that. But this is so what I'm showing you in this slide is actually a snapshot from a molecular dynamics simulation where you have the lipid membrane, and so here you see that this is, you can consider this as the outer leaflet, okay, and this would be the inner leaflet, all right, and these cyan shaped molecules are nothing but water molecules, and here also you have the water molecules. And what is interesting to see that, you just you can take a look at any of the lipids, right? So for example, in this figure, there are some interesting features, okay. So for example, you see that so as we were discussing, right, so that, although when you draw the lipids, you draw it in a zigzag manner to depict the most stable staggered conformation, but you can see that many of these lipids are not, although they're pretty, they're saturated, they're not present in a fully zigzag manner. For example, this one here, right? So you have ZZ configuration. Suddenly you probably have a cis configuration, although it's difficult to say here, if this is a cis or a trans, but here you have one, for example, here also you have another one. Also could be that this is a cis one, and you can see the kinkedness of this cis lipid, because we spoke about these lipids and the saturation, and you might want to remember that these lipids have unsaturated centers, typically 9, 10, and 12, 13 or something, and where these unsaturations in naturally occurring lipids are cis, because cis induces a much stronger kink than the trans, right? And yeah, so for example, also here so the lipid which is drawn here in green is actually taken out of the environment and shown in here, and you can see that there are so even for these, so this is the one which is present in the inner leaflet. Oh, what happened to my pointer? Oh, hold on, can you still hear me? Hello?
So I had a
Yes,
question
sir.
in
Yes,
this
sir.
section.
Yes, sir.
Okay, my screen seems to be frozen. Okay, yeah, ask your question.
Sir, good morning, sir. My question is that in the lipids,
Yeah.
why aren't the double bonds conjugated?
Why are they not conjugated?
Yes.
Well, I don't know. I mean that. I mean, do you envision any specific advantage to have them in a conjugated manner than in
There
a
is
saturated manner?
the only particular advantage that might be is that uh, uh, like it might just increase stability or something, but I can't think of any other reason.
Stability in terms of, you mean rigidity? Is that what you mean?
No, I mean in general, like the stability of the double bonds. I mean robustness of
Sir,
the
like
double bonds
resonance
to
stability.
accidental reduction or something would be more because it would be more stable.
Well, I mean, see, it's uh, well, accidental reduction. I'm not sure that whether in our nature we do have hydrogen, but we don't have nascent hydrogen that will spontaneously reduce these double bonds, because see what I'm talking about. For example, you see that in the naturally occurring lipids, you just have one double bond, the cis one, right? If there is a possibility of accidental reduction, I think one is a lot more vulnerable than multiple, isn't it? But despite that, you
Excuse
still
me,
have
sir.
one, isn't it? Because it's
Sir,
lot
like,
because it's a lot more energy rich process to have multiple double bonds than one, right? So if the job is done with one, it's a nature is sort of pretty lazy. It optimizes solutions, okay? And the way it optimizes solution, or the way it has optimized solutions, is by evolution. And that's how probably there were I mean, nevertheless, I mean, we will in the next slides. When we come to the next slides, you will see that there are molecules present in the lipid bilayer which have conjugated double bonds. It's not that they are not there, but yes, I mean, I understand you have a concern that it could be much more stable, but at the same time, it's a lot more energy rich. And the other thing that you would have is, see, if you have multiple double bonds, you can clearly understand that you have, since you have the multiple pi bonds, there will be the canonical forms, right? Isn't it? So, for example, 1,3-butadiene. So, 1,3-butadiene has the canonical form when the double bond is also between the second and the third bond, right? Not just the 1, 2, and 3, 4. So that means that if you have conjugated double bonds, the single bonds also have partial double bond character, and that will induce enormous amount of rigidity into the lipid chains. And if that happens, the flexibility of the hydrophobic core is sort of reduced. And if the flexibility is reduced, that will well, in another terms, it means that it's more rigid. And if systems are more rigid, they will probably be less dynamic than than the ones where you have mostly saturated carbon tails. Do you understand what I'm saying?
Right. Yes, sir. Yes, sir.
Yeah. Hold on. I have to. I don't know why that happened, but anyway. Okay. Yeah. So can you still see my screen? Can you all?
Yes, sir.
Okay,
Yes,
good.
sir.
Yeah, good. Okay, fine. Yeah, so here, so here this one is the lipid which is present in the inner leaflet, and this lipid is present in the outer leaflet, and you will see that there are multiple. For example, we don't think that you can count. This site is definitely not the specific site for the formation of the cis double bond. This is 9, 10 or so, but this will be probably third or fourth residue where you have the cis double well, cis conformation, right? So it clearly demonstrates, although this is a molecular dynamic simulation, but it sort of very well replicates what is present in the real nature. I will just tell you in a second, how you can demonstrate that. So it means that lipids are very, very flexible, and you have translocation, meaning that for example, in one snapshot, when you are seeing this specific green lipid at this position, and another time, this green lipid probably is not stuck at this position. Okay, it is actually freely floating, and it goes from this position to that position. Okay, and there are very nice experiments which demonstrates that. But nevertheless, one thing you must you might want to see, or you can probably see in this, that in this yellow region, this thick yellow region, which is the hydrophobic core of the lipid bilayer, you don't see this head groups, right? Because to have the head groups insert into the hydrophobic membrane is enormously energy rich, right? So energy rich meaning that, for example, you have to desolvate the phosphate groups to have it inside. And that desolvation energy is enormously high. All right. So that is why, you can, well, now you probably can envision that, why it is very difficult. The architecture itself prevents the passage of molecules which are pretty polar. Okay. Whereas, if you consider hydrophobic molecules have higher chance of passing through the membrane, but nevertheless, one also has to understand that, it's not that hydrophobic molecules can pass through easily, but the first thing is hydrophobic molecules have also to first interact with the head groups, and for that interaction you have to displace the water molecules. And many of the cases, it's known that there is a very thin layer of water molecules which is extremely tightly bound to the polar head groups. Okay, so that is also energy rich. So it means that for molecules to pass through the lipid bilayer, you have to have certain balance between the polarity or non-polarity, meaning that the molecules have to be amphipathic. Otherwise, if the molecules are very hydrophobic, that does not necessarily mean that it will pass through. Or if the molecules are pretty hydrophilic, it doesn't mean that the molecules will pass through. Okay. All right. So as I was saying that, so there are very nice experiments which actually can demonstrate that the lipids actually freely are free to diffuse in one specific leaflet, and this is this experiment where I'll show you the data in a minute, but just first understand the experiment. So essentially, what people have done is. Uh, for example, what they have done here. Hold on a second. I mean, let me. Uh, oops, oops. Just a second. Just hold on a second. Okay. Yeah. Sorry. Here where am I? Yes. Here. Yeah. So laser pointer. Yeah. Yeah. So what was done is it was a beautiful experiment where can you all hear me?
Yes, sir, we can.
Yes, sir, you are audible. Yes,
Good.
sir.
Yes,
Yeah.
sir, you are audible.
Okay, thanks. Yeah, so what one can do is actually, there are proteins which are fluorescent proteins. You might have heard of such proteins. For example, there is one fluorescent protein called as green fluorescent protein, which is obtained from the jellyfish. Okay, so what one can do is, thanks to the cloning techniques, you can actually attach green fluorescent proteins, which is shown here in the shape of eggs here, right? You can attach these green fluorescent proteins to the lipids, all right? And so when you are presenting green fluorescent proteins to the lipids, they were sort of exposed on the surface of the lipid bilayer, right? And they're sort of freely moving. Now the whole goal of our experiment is to demonstrate that lipids freely diffuse in the membrane. Now how do you, so that is the question that we are asking, right? That it will show that the lipids are basically dynamic and they are not sort of rigid and static in terms of their three-dimensional coordinates. Okay, so yeah, so this is the experiment where people have actually tagged green fluorescent proteins to lipids, and then what you do is a beautiful experiment where you can take a microscope, and this microscope is attached to a laser, and using that laser, what you do is you focus at a specific point, and at that specific point, what you do is you shine the laser at a specific wavelength, which well, in simple terms, it destroys the fluorescent property of these molecules. Okay, so this is what is done in the real experiment, where so here in this graph, what you see that you have the fluorescent intensity, okay, in the y-axis, and in the x-axis you have the time in seconds. So you can see that there are several well, each dot sort of represents each each green fluorescent protein, and what you can see that there are many fluorescent proteins, and which actually have a very high intensity, right? And then suddenly when you shine the laser, what happens is, I mean, you bleach them, meaning that you destroy the fluorescence of these proteins. And since you destroy the fluorescence of this protein, and your objective is specifically focused at this point, okay, only at this point, you're not moving your focus, all right? And what you see that, once you shine the laser, immediately fluorescent intensity drops, right? And after the fluorescent intensity drops, what you can see that, since you're continuously monitoring the fluorescence at this specific point of focus, you see that gradually the fluorescence sort of regains. Okay, so this kind of experiment is called as fluorescent recovery after photo bleaching, FRAP experiment. It's done for many different kind of experiment, but yeah, so what does this kind of experiment demonstrate, right? So now this floor is open for your suggestions. What do you think? What is actually happening?
So why they regain the fluorescence again?
Correct. That's what I'm asking. What do you think?
Because
I think, sir, probably it is diffused. Probably the lipid molecules diffused throughout the membrane.
Yes, yes. And what about how? Why do you say that they diffuse through the membrane? Why not these molecules which have become whitish? They regain their fluorescence.
Because if that had
Because
happened,
it
the
is
intensity would rise to the
destroyed.
original 100%. It would have risen to like
Yeah,
that
yeah.
600 units. But there is
It
a
is
certain
not.
deficit that happens.
Yeah.
A
Why do
certain
you,
percentage has not returned to the top.
why do you think so?
Like we can see
Sir,
the
from the graph,
like
we can see
the
from the graph.
intensity goes back to like it stabilizes at a lower value.
No. I understand that. But my question is, why do you think that if these were the molecules which were bleached, right? If they regain the fluorescence, why would it come to the same level?
No. I mean, it should, right? I mean,
No, that's what I'm asking. What is your thought process behind it? I mean, why
Sir, I
would it?
think I think the molecules did not regain the its fluorescent properties. It just distributed all over the place. As we can see in the third picture, that
Hmm.
there is only one white fluorescent particle, and
Right.
others moved away. So
Correct.
it kind of like distributed equally.
No, I understand. But my question is, why do you think so? Why not? Why
Uh,
not that? Why didn't that the white ones regain their fluorescence?
so
I'm asking for an
I can
alternate explanation.
argument for Prithvi's whatever he said. Like, so I would say that each molecule is similar to each other. So if one of them can regain the light, the color, then all of them should be able to regain the color. But if all of them are
Sir, we can put it in this way: like once it has regained a certain level, we
Hmm.
can photo bleach it again and again until, and we'll see that it keeps rising to a lower and lower level.
Uh,
Yes, and
Sir,
that will say that they can't regain that property.
no, fine. This. Okay, so we will come to that in a minute. But my first question is, why do you think that there is some sort of a how do I say, it's a transverse kind of diffusion than the molecules not gaining the fluorescence, meaning that if there is a
if we
population.
observe a different if we observe a different portion of the membrane, then we can see that there also the intensity has decreased, which means that some of the bleached molecules would have gone there.
Sir, that
sir,
that
can I
will
ask a question, sir?
that will be difficult to do. Yes, you can ask me the first. I am asking the question. I need an answer,
So
explanation.
we can see
No,
from the
sir, I want to say that is it really possible for the bleached molecules to regain its fluorescent properties?
That's a good question. That's a very good question. The answer is yes. It is possible. Okay. Now, well, I'll give you the answer. So it is possible. Okay. This is something that the information which I have not given you. It's very much possible for the molecules, these whitish molecules, to regain their fluorescence, but it turns out at which the time scale at which these guys retain or regain their fluorescence is much longer. Okay. All right. So it's not within seconds, right? It's within minutes, or it could be hours also. So the answer is actually hidden in this time frame. Okay. So this time frame, this so fast recovery after the bleaching, it actually indicates that it's not those molecules regaining the fluorescence who have actually got bleached, but instead some other molecules would have come in the point of focus and replaced them. Okay, followed.
Yes, sir.
Are there any questions? Okay, and the other thing, I don't have a direct answer, but I believe what is happening is uh, well, how do we put it? So, I do not know why actually this is low. Ideally, it should be high. Okay, unless it's some sort of a artifact of the measurement. That is why it's initially it was at 600. Because it's also possible that okay, so let's say that when you are bleaching, although we are saying that you are bleaching at this specific region, the circle, okay, so the laser, and it must be a very good laser, but I am not sure that the thickness of the laser, if this is the specific region, up to angstrom lengths or up to whatever nanometers length at which the molecules are bleaching. It is also possible that some of the molecules in the periphery, which are outside this circumference, they are also bleached. Okay, so that is one possible explanation that why the molecules which move into the field from outside, okay, they are not regaining the fluorescence back to the original level, because some of them might have also lost their fluorescence properties.
So when new molecules replace the bleached ones, how can we say that the previous intensity will be retained, because new ones are coming and their intensity may be different? May
Yes,
that also a reason for that?
that's what. Oh, okay. So you are saying that the intensity of the molecules, the fluorescent intensity of the green fluorescent protein, is that what you're saying?
Yes, sir.
You mean that the initial ones? Oh, you're meaning that this population's intensity is higher than this population's intensity. Is that what you're saying?
Yes, sir.
No, but no, see, well, it's quite likely. But what we are considering here, you see that, this is probably a mean intensity, right? So the intensities are different. You can see from the individual dots, right? Some are low, some are high, but you have a mean intensity. And here, what you see that the mean intensity itself is quite low. Forget about, the specific intensities. Do you understand what I'm saying? We are talking about the intensity of the overall population.
Yes, sir.
Yeah.
So why does the intensity become zero?
Why does the intensity become zero, or why does not?
Why does not?
Oh, I guess this is just, there is something called as autofluorescence. Okay. So, and again, it has something to do with the, because, it's not that it becomes completely dark. Okay. So when you're looking through when you will look through the microscope, I don't know, second year, third year in a confocal microscope, you will probably see that, after this bleaching experiment, it doesn't really go to darkness. Okay, so that is why it's not zero, because there is still some autofluorescence. Well, and that autofluorescence might come from because you are not doing on a surface, right? There is a buffer there, right? There are salts present in the media, and there are molecules. Although you are specifically looking at maybe 420 channel, 430 channel, whatever, because of the green fluorescence, there are bleed through from other proteins which are also fluorescent, not in that region. If you read about fluorescence, you'll see that there is overlap, and that is why it doesn't really go to zero. It's just autofluorescence. It's nothing to do with the real fluorescence of the molecules. Okay, does it answer your question?
Yes, sir.
Okay, all right.
Sir.
Yes.
Sir, how can we be sure that it is not the protein molecules which are migrating on the lipid surface and the entire lipid molecules migrating?
It's actually the entire lipid molecule which is migrating.
Sir, so how can we be sure? It could be that the proteins are just diffusing and the lipid layer is stable, beneath.
Well, proteins, what do you mean? So what we have done here is we have the green fluorescent proteins. They are tethered to our lipids through a covalent linkage.
Okay, sir. So means that linkage is strong.
Yes. It's not non-covalent linkage. Okay. So, so here, you don't have a population of protein which are freely diffusing, and then they're non-covalently or not bound to the or not tethered to the lipids.
Thank you, sir.
Okay, anything else? All right.
Sir,
Yeah. Yes.
I think one reason of decreasing of intensity,
Hmm.
like there are some bleached molecules which do not go away from the circle because distribution is random. If there are some bleached molecules, they must remain in the circle.
That's also possible, yes. So that's why you're saying the average intensity is low?
Yes, sir.
It could be. It's possible.
Sir,
I
it
mean,
is
it's
also
possible. It's possible depending upon, so for example, see, this is, I don't think it's a single molecule kind of experiment, because if you are to sort of see these green dots, actually, you can, for the time being, you can understand, or imagine them as a single GFP molecules. Okay. So what your colleague is suggesting, that the distribution of the bleached and the unbleached molecule in a specific area of interest, right? That's what you're saying.
Yes.
Yeah. Yes, exactly. Yes. So in a specific area of interest, probably there were 100 molecules. In the specific area of interest, there are probably after bleaching, maybe there is a 80-20, and that is why probably it's low. It's yes, it's very likely possible. Absolutely. Okay. All right.
Sir,
Good.
but after some time, once it has regained the fluorescence,
Yes.
I think it should again reach the value of 100.
You mean the 600?
Yes, sir, because once the fluorescent protein which is bleached regains its fluorescence, it should again reach the full value.
It's possible, yes. So that will be a very long time scale then. Yes. So if you keep on measuring for a long time, then you will see it. But there are, okay, so I'm telling you details which you may not know now, that
Sir?
what happens is, when you keep on measuring, the way you measure is, measure the fluorescence is that you excite it, and the excited molecule emits a light that you detect, right?
Sir?
Yes,
Hold on.
Yes.
So what happens is, there is something which is called as bleaching. I mean, here it's what you're doing is, you're doing a forced bleaching. Okay. There is something which is called as spontaneous bleaching, meaning that because of the laser intensity, no matter how low it is, okay, some molecules tend to lose their fluorescence properties. Okay. So it's just a technical problem, that even if you measure for a long time, you may not achieve this. That has no consequence on the conclusions of the experiment, but it's just that the molecules have not. They're undergoing sort of very slow bleaching. Followed?
Yes.
Yes, sir, got it.
Forced or spontaneous bleaching then?
Sorry?
I got confused whether this is forced or spontaneous.
No, this is forced. This is forced.
Forced bleaching.
Yeah, this is forced. That is why it goes to, I mean, within fraction of seconds, it goes to very low intensity. Okay.
Sir, and in the previous slide, I had a doubt. What if the head group of the lipid was neutral, like the phosphate as well as was negatively charged, and we had a positively charged moiety attached, so the net charge would be 0.
Yes.
So then, would the thin layer of water be there even when the head group is neutral?
Yes. Is water charged?
No.
So now, but if there is a water molecule, are there surrounding water molecules?
Oh, yes, okay.
Okay. All right.
Sir?
Yes.
Sir, I have an explanation for your previous question. That why don't we think that these molecules regain fluorescence? So like that, initially in the bleached region, there will be all bleached molecules.
Correct.
So the rate of the diffusing will be proportional to their number. So
Yes.
we can see from the graph, this graph looks like a first order chemical kinetic reaction.
Okay.
But if they regain fluorescence, the type of graph would be not like this.
That's fine. Yeah, sure. See, yes, one way is that, but no, but how do you know? How do you know
Yes,
that?
even if it had been due to the GFP regaining fluorescence, it would still have been a first order graph, first
Yes,
order kinetic graph.
it all
Yes.
depends. It all depends upon the time scale.
Yeah, the time scale would be different.
Yes,
Yes, yes.
time scale is what is very important. You probably will see a first order, second order. I don't know what order you will see, or you maybe just see a linear kind of correlation. I don't know. It all depends upon the time scale. Maybe this will be in hours then. Okay. All right. So let's go to the next slide. So yeah.
Excuse
So
me,
what?
sir.
Yes.
Sir, I don't get the answer to that question that someone asked that how can we be sure that it is the lipids that have been diffused and not the whole bilayer itself that has delocalized?
Whole bilayer.
Like the pink color layer which is
Yes.
indicated here, it is moving. It is also possible like that.
I I do not understand. What do you mean? The whole bilayer is moving, meaning that the whole chunk of this molecule is moving. Is that what you mean?
It is possible that the this tails and head lipids which are shown in the diagram are
Yes.
fixed to this bilayer pink color one,
Okay.
and the bilayer itself is moving so that the area on which we have bleached the proteins
Hmm.
will be on the out of focus of our region.
Okay, good. How will the bilayer move without the movement of the lipids?
Uh, so the bilayer itself is constituted only of the protein lipids.
There are proteins also.
So isn't it possible that the bilayer itself moves?
Unless the cell moves, if that's what you mean.
Yeah, that's a possible thing.
Yes.
Then how
That
can we
is
be sure that only the lipids have diffused in and out of the
That's
focus
a good
region?
question. It's a very alternate. Yeah, yeah, you're right. So essentially what is done in this kind of experiments is the cells are tethered to the glass slide. Okay, this part what you see. Okay, though there is something called as polylysin coating. So that means that cells are, they are sort of adhering to that region, meaning that so in that case you will not have that the field of focus has completely shifted. Do you understand what I'm saying?
Yes, sir, I got it.
Yeah, that's a good alternative. Yes, I like it. Yes.
So
Very
the upper part of the bilayer is the polar part of the lipids. So how can it move differently?
No. What he is trying to say is, what if the entire block, right? So what we are doing here, it's cut into specific XY. Okay. So imagine it's a two-dimensional block. It looks like a Kaju Katli. Okay. So imagine there is another Kaju Katli next to it, and this Kaju Katli sort of slides on your left, and another Kaju Katli comes in the field of view. If that happens, then also you will have a similar behavior,
Okay,
right?
sir. Yes, sir. I now understand.
Okay. Now, but what if we tethered the Kaju Katli onto the surface of the glass slide? If we do that, that will ensure that there is no movement of the Kaju Katli. Correct? Yeah,
Yes, sir, this is
Yes, sir.
exactly what I
that was a
mean.
good question. Yes, good. All right.
Sir, I have a question. Hello.
Yes. Tell me.
Sir, if we repeat the experiment in the same way with proteins, like if we combine the fluorescent molecules with some of the proteins and then repeat this laser type experiment, then the result will be the same or different?
I don't follow what you mean. Do you mean that?
I mean, sir, if we connect some of the fluorescent molecules with some protein molecules,
Where are
and
these present now? They are also present in the membrane.
On the upper side of the membrane, that membrane which is not attached to the glass slide.
Okay. Okay. So would you see a similar kind of graph?
I want to know, will there be a similar kind of graph or the result will be different?
Well, I have not done this experiment, but I assume that it will be different. I mean, because see, lipids are pretty small molecules. Well, and you will see why is it different. I mean, at least theoretically, you will see now in few slides. What I assume is the motion of the proteins are relatively restricted in the membrane as opposed to the lipids. Okay, so if the motion is restricted, you would probably see a recovery, but it will be a much slower recovery. All right.
Okay, sir.
Okay. So let's go to the next slide. So, I mean, just for you to know that there are not only just lipids and cholesterol kind of molecules present in the membrane, there are also other forms of lipids. For example, those are called as vitamins. Like so here, so you have so there are water soluble vitamins and there are also fat soluble vitamins. Now now the molecules or the vitamins that are present in the membrane are typically the fat soluble ones. For example, you have vitamin A, vitamin E. This is a vitamin E. You have vitamin D3, vitamin K, and so on and so forth. And just to remind you, so one of your colleague asked that, what if, there are conjugated molecules? You can see that, see, this vitamin A guy, I think it plays an important role in your eyes and all. Maybe I think it does. It does. How? I don't know. So this you have this conjugated double bond because it's associated with the chromophoric properties of these molecules. So you do have the presence of vitamin A, but vitamin A, definitely this molecule is not predominant in the membrane. There is some percentage of this molecule present in the membrane. I don't know what exact percentage and which membrane. Most plasma membrane is present. Likewise, there are also other molecules which are present in the membrane. For example, these are, polyisoprenes, right? So probably what are isoprenes, right? So it's what 2-methyl-1,3-butadiene sort of, okay? And you can polymerize those, and you can get these kind of molecules which are called as farnesol, geranyl, geranol, dolicol, okay? And here you see that here you do have the presence of trans double bonds, right? Now, one thing that is that I must want to tell you here that there are, for example, proteins which let's say that they have for example, there is a protein which has some role in the cytosol, but once the role is done, probably it prefers to stay anchored to the membrane. Okay, there are many such protein. One such protein is RAS. Okay, so don't worry about what RAS is. You will probably study about it later. There's one such protein. Okay, there are many such proteins, but the question is, how do such proteins get anchored? Which are cytosolic protein, meaning that you can imagine that if a molecule is cytosolic, that means it must be sort of globular and soluble. Okay, and it does its job in the cytosol, and suddenly once the job is done, it goes and hangs to the membrane. Okay, or the other way around also. If, for example, if a molecule is to be hung onto the membrane, so imagine there is a protein, okay, and or imagine there is a molecule, that is freely rotating in the cytosol, and when the job is to be done, that molecule undergoes some modification that that sort of tethers that molecule, anchors that molecule onto the membrane. So in this previous experiment, what you saw is, so it was genetically done. It was sort of when you go to the lab, you learn the cloning techniques. You yourself can do this. That you can do some little bit of chemistry, biology, molecular engineering, molecular cloning, whatever you call it, and you can present, or you can generate molecules with protein lipid fusion. So what I want to say here that this is something that is also done by the nature itself, right? There are proteins which are actually farnesylated. right? Farnesylated meaning that there are proteins, and those proteins undergo. Let's try to do this. Uh, let's see if this guy works today. Uh, I'm trying to get my whiteboard. Let's see if this thing works today or not. Uh, okay, yeah. Okay, so what I'm trying to say here is, so this is the membrane. Okay, all right.
It's not
There
visible.
is not visible.
Yes, sir.
Just visible.
Sir, it is
Sir, it is
visible.
visual.
We can see it. Sir, there is some time lag, I think.
Yeah, probably there is a lag. Yes. So see, what I'm saying is, yeah.
Yes, sir. It's visible, sir.
Okay, okay. Now what happens is there is a protein. This is protein P. Okay, and what happens is there is this that farnesol molecule or whatever. Okay, so you have that double bond and you have the OH groups. Okay, so what happens, when the protein is to be anchored onto the membrane. Now this is our membrane. Okay, so this is please don't write on the board. Okay, uh, all right, so what happens is, so there are proteins on the C terminal of which there is something which is called as a CAX motif. C, A, and X. Okay, now what is CAX? CAX, as you will see later, C is an amino acid which is called as a cysteine. A and A, these are aliphatic amino acids, and X is any amino acid. Okay, so there are some proteins which have this tetrapeptide sequence at the C terminus of those proteins. Now, what is C terminus, N terminus, we will see later. But for the time being, just imagine that there are proteins which have tails having this CAX motif. And cysteine is an amino acid where there is a thiol group, SH. Okay, sorry about my writing. This is a SH. Okay, now these molecules, these farnesylated molecules, they are actually in the form of a phosphate group. Okay, now what happens is, when the job is to be done, when the protein is to be anchored onto the membrane, what happens is there are specific enzymes called as farnesyl transferases. What these farnesyl transferases do is they put this farnesyl molecule, farnesyl phosphate molecule, onto the protein. Okay, it's a very simple kind of nucleophilic kind of reaction where you have the sulfur, the thiol sort of attacking the sorry, the O. This would be the CH2 here. It cannot attack here, certainly. Where is my? So this guy, you have the lone pair, it directly goes and attacks this carbon, because phosphate is a very good leaving group, and this phosphate guy goes right, and then essentially what happens is the protein gets modified with the farnesylated molecule. Now once the protein is farnesylated, there are few events that takes place which replaces this AAX. Okay, and the molecule or the terminal or the tail of this protein becomes pretty hydrophobic, and then it of course does not like the aqueous environment present in the cytosol. It just goes and anchors into the membrane. Okay, and there are so nature also exploits this kind of engineering via very specific amino acid sequences. All right, so yeah, and so that's how proteins which are for example, if they are to do specific job onto the membrane and shuttle between the membrane and the cytosol, that is how they take advantage of this CAX motif, and these series of sequences where first it gets farnesylated, and then the amino acids are essentially removed from the CAX motif, these three amino acids, and then the molecule essentially goes and goes and gets tethered onto the membrane. Okay. That's how these okay, so hold on, I have to share my screen again. Yeah, so Yeah, so that's how these molecules are very useful, like farnesol, geranyl, geranol. Okay, so they help in tethering proteins onto the membranes. All right. Now, there are also specific regions. Let me finish, and then you can ask the questions. Is your next class recorded, or is it a contact class? I mean, like on live class?
Recorded.
It's recorded.
It's recorded.
Okay, then I will just finish the lipids. I have just few more slides. So there are also specific regions in plasma membrane. Those are called as lipid rafts. Okay, so plasma membrane, it is not very homogeneous. Okay, homogeneous in terms of both the composition and the thickness. So you can see that in this part the thickness is less, and then this part the thickness is more, and these parts are called as lipid rafts. And in this lipid raft region, there are many other things. For example, you have for example, there are proteins which are called as GPI anchored proteins. We'll see later what are GPI anchored proteins. And you have the presence of several other proteins. So this, in this green loop, this snake kind of thingy is actually a protein. You have lots of cholesterol present in this part. Okay, and people have realized the presence of lipid rafts because it turns out that unlike the other part of the membrane, which is sort of, you can extract it using a detergent, a mild detergent. There are certain regions which are very difficult to solubilize, right? And those regions are called as lipid rafts. That's how people initially realize that, oh, there are certain regions in plasma membranes which are sort of very thick. And then eventually, I mean, lots of work is still being done to identify what are typically present in lipid rafts and all. And now, I just want you to highlight, one of your colleagues asked that, what if there are proteins, if you tether green fluorescent proteins onto a specific protein that is present in the membrane, what will happen? So yeah, so you will see that many of the proteins are present in this raft region, which are very rigid, meaning that the unlike the non-raft region where you have relatively larger motion of the lipids, right? Whereas in these regions, the presence of the cholesterol makes these regions very, very sort of rigid. So obviously, diffusion is a lot less than the regular regions. All right. Okay, so this is lipid rafts, and you can also using very nice techniques called as atomic force microscopy. You will probably study about that later, or just read something on the net about atomic force microscopy, AFM. And using AFM techniques, people have been able to show the presence of raft. Okay, now this is not a real cell membrane. This is sort of a it's a surface bound membrane that has been artificially constructed, where so they essentially look like sort of icebergs in a sea. Okay, so this sea is nothing but you have the regular lipids, and it's known that depending upon the composition of the lipids, cholesterol and all, these guys tend to cluster. Okay, and this clustering is leading to the formation of this red icebergs, and these are the lipid rafts. I'm not sure whether still today people have been able to visualize rafts in real membranes. You can look it up and let me know. Okay, but this is a supported membrane, meaning that it's an artificial membrane which is made from using the compositions of the lipids that are typically present in a plasma membrane. Okay, all right. Now, yes. So, well, we will talk about, this is a GPI anchored protein, but I'll talk about it when we study the carbohydrates also. Okay, the other thing, so other thing that is important for you to know that, is, for example, lipids. So imagine like we saw that, so if there are proteins that need to be shuttled between the membrane and the cytosol, there have to be specific modifications of the protein. For example, in the C-terminals part of the protein, which is a CAX motif that gets farnesylated. What about the lipids? For example, if the lipids need to be transported from one part of the cell to the other part of the cell, or from the cytosol to the membrane, or vice versa, how does it happen? Because then probably it's a lot of energy to actually do some chemical modification on a specific lipid, and undo that modification to again make it more hydrophobic. So, nature has devised very unique mechanisms. For example, there are specific transporters. For example, this is a protein, and that actually transports lipid molecules. For example, this protein, it sort of jackets the oleic acid, and now this is a soluble protein. In the core of the protein, the oleic acid is present, and it can happily mask this guy and take it from the membrane to the cytosol or wherever it is required. For example, maybe from the plasma membrane to the membrane of the Golgi or nuclear membrane or something. Okay. Likewise, there are also proteins which are called as, for example, which transport cholesterol. Okay. So a cholesterol, because cholesterol is also, it's involved in many other signals, besides being structurally inducing some sort of a rigidity of the membrane. So there are cholesterol transport proteins. For example, what you can see that, in this red backbone, this is a cholesterol molecule, and there is another cholesterol molecule present here. Okay, along with the cholesterol, there are also certain phospholipids, and there is a cholesterol transport protein which sort of again jackets both the cholesterol molecules along with a couple of phospholipids and can shuttle this protein, because the protein outside of the protein is pretty polar. Okay, so this protein can transport these guys from the membrane to some specific parts, organelles, compartments in the membrane. Okay, likewise, you know so there are proteins which are called as apolipoprotein. These are crazy guys. Okay, so for example, apolipoprotein in the apoform, meaning that when it's not bound to lipids, it has a globular structure. You will see later what is a globular structure. Imagine, this is a very nice compact structure of this apolipoprotein A1. But when it has to do its job, what it does, it sort of undergoes unfolding. Okay, meaning that but some changes happen which releases or makes this molecule pretty loose, and what it does, it forms a disc kind of structure. So imagine, yeah, so this is the side view, this is the top view. So imagine you have a coin. Okay, so 1 rupee coin or whatever, and the periphery of the 1 rupee coin is the protein. Okay, so you're looking at the coin from the top, and the coin is essentially filled with all sorts of lipids, right? And this is the sidewise view of the coin, where you see that the edge of the coin is essentially the protein. And of course, it's imperative, you can clearly understand that obviously the protein sort of forms the circumference or the periphery, because these protein, the inner side of these, this helix, as you will see later what a helix is definitely composed of hydrophobic amino acids. Because if you have polar amino acids which is facing the disk of the lipid, obviously that won't be stable, right? So that is why, these protein, also in this case, the inner part of the amino acids or the helices are actually pretty hydrophobic amino acids. And that is why it can form a very nice circumference around this disk of lipid, and transfer, so it's amazing, right? It actually transfers a chunk of lipid from one membrane. For example, if the cell realizes that, I need a chunk of lipid from my plasma membrane to my endoplasmic reticulum or Golgi or maybe nuclear membrane, it can actually shuttle this chunk of lipid from one part of the cell to the other part of the cell. The reason is because this protein, the outer part of this, of course, you see that the top part and the bottom part is, of course, this polar residues, right? So there is no problem in getting it dissolved into the cytosol or in aqueous condition. And the disc, it sort of jackets, so the amino acids that are present, it sort of jackets the hydrophobic part, so that there is no real energy requirement for the solvation about this. And obviously this membrane or this protein is composed of amino acids, and from the outer surface, the outer part of this helical protein is composed of amino acids which are polar, but the inner part is composed of amino acids which are hydrophobic. So this is an amphipathic helix. Okay, and that is why it can actually do its job. Right. So that is the end of lipids. Now you can ask me any questions that you may have. So in the next class, we'll start with glycans.
Sir, I had one question about the CAX motif. So in that the C terminal is at the cysteine or is it at the X?
Oh, it's at the X.
Okay, yes.
Because
So the C terminal is somewhere like just a few a few amino acids short of the end of the protein.
No. I mean, no. The C terminus. So if there is a large protein, there is a C terminus and there is an N terminus. Okay. So what I mean to say that this tail is present at the CAX motif is present typically at the C terminus, not at the N terminus. We are not specifying that which amino acid is the terminal amino acid.
Oh, okay.
But essentially, what happens? There are modifications after it gets farnesylated. I think there is proteolysis that leads to the deletion of that, or not deletion, but the hydrolysis of that CAAX. Okay. So the C becomes the C terminus, and then there is probably some sort of a methylation or esterification of that, and then that terminal, because it's also, see, if the C terminus typically in proteins, it's a free carboxyl group. Okay.
Yes.
But if there is a free carboxyl group, that is also heavily solvated. So that also has to be made very hydrophobic by esterification. Once it's esterified, then that that tail present with the side chain of the cysteine, which is the thiol, can happily anchor onto the membrane.
Okay. Yes, sir. Okay.
Are there
Excuse
any
me,
more?
sir.
Yes.
Sir, so in the CAX, like the the protein one, how does it bond to farnesyl? I couldn't understand how the sulfur of cysteine gets added.
Okay. 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 So a phosphate group, right?
Yes.
Okay. All right. Good. So I have ChemDraw here. It might take a little long to do it. So okay. So what happens is methanol.
Yes.
Okay. Methanol, right? So imagine methanol is it forms an ester with a phosphate group, right? Methanol is an alcohol, and phosphate or phosphoric acid. Okay, it's an acid. So you can form this essentially this CH3. Okay, I mean it's very OPO4. No, I can't. I mean, I would have preferred board. So essentially, what happens is you have CH3OPO3, right? Now you have to imagine.
Yes.
Right now, so you have CH3OPO3. Now that's an ester. Okay. Now, if you have a nucleophile which is in proximity, what happens that a nucleophile can attack the carbon, the C of the CH3, and OPO3 can happily leave because it's a phosphate group, right? And since it's a phosphate, that negative charge is very well stabilized by the 3 other oxygen that is present on the phosphorus, right? So that is a very good leaving group. So this is exactly what happens in a farnesylation. So that farnesyl all that my slide is still shown, right? Here. So can you let me put on my laser pointer. Yeah. Do you see my laser pointer?
No, sir.
No.
Really?
Yes, sir. Now
Yes, sir. Now we can see.
Yeah. So what happens is you have the OH group, right? So this OH group gets phosphorylated. Okay. So you have OPO3. All right.
Yes. Now I got it.
Yeah. Now the cysteine, that SH of the cysteine, it's a reasonably nucleophilic species, and that undergoes an attack at this carbon center. So this OPO3 leaves, and this S gets farnesylated.
Okay.
So is the farnesol with the phosphate inside the inside the cytosol, it is in this phosphorylated form, and when cysteine attacks, the phosphate
Yeah.
groups goes away.
So there are specific enzymes, I mean, these are all sort of linked to each other. It's not that randomly things are happening. Okay. So because this reaction, what I told you just now, it is catalyzed by a farnesyl transferase, right? So because for example, you have to have a pocket where both the farnesyl phosphate and the protein, that protein with the CAX motif binds, and then this reaction can take place. Because cell is basically a soup or a gelatinous gel kind of thing, and the proximity of, because these are very specific reactions, the proximity of the two reacting species is very important. So enzymes are nothing but catalysts, or let's say these are dyes, okay, which brings these two, the reactant, both the reactants in close proximity, and then promotes this reaction. This is something that you have to understand, that cytosol is basically some horrible glue. It's like a gelatinous material. It's a gel. It's not like liquid and freely floating.
Excuse me, sir.
Yes.
Sir, I had a question regarding the fluorescent proteins and the conclusions we drew.
Yeah.
So we were claiming that this is due to the diffusion of the lipids by looking at the time scale over which the regain of fluorescence occurs.
Yeah.
So to compare it with how long it typically takes for a single protein to regain fluorescence, we need to create we need a previous experiment in which we have static proteins and then we measure. So my question is, how do we create a static arrangement of proteins for reference of time scales?
Okay, so that's not very difficult. See, it's very simple. What you could do is, you don't even have to create a static arrangement. You can take a tube in which you have the green fluorescent protein present, okay, and just in a solution, and you just bleach it, okay, and monitor the recovery, how long it takes to recover. You don't have to worry about the static presentation. Do you understand?
So is the energy enough? Like, does it require high energies for like a tube level of bleaching, or is it sufficient?
No, not necessarily.
Okay.
No.
Okay.
It's a very simple experiment. You can do it in a cuvette itself.
Okay, sir.
Yeah. Because the only reason why we can do it in a cuvette, because it's I don't know, but I believe it's in hours.
Okay, sir.
Excuse me, sir.
Yeah.
Sir, when you showed the lipid raft, so the lipid raft part of the bilayer, it was very ordered, whereas in the other parts shown very disordered, the lipid tail. So why is that so?
Yeah, I believe it's because of the cholesterol. Because so cholesterol, you would have seen that cholesterol is composed of this what is this called, perhydrocyclophenanthrene ring, right? You have three fused ring systems, right?
Yes, sir.
So since you have three fused ring systems, that is extremely rigid, then if you look at cholesterol from sidewise manner, it'll sort of look like a rod. Okay, it will not move, as opposed to a free carbon tail, right? Since you have the presence of cholesterol, what happens is, and then the presence of these freely moving lipid tails, it sort of restricts its motion, right? It induces some sort of a, it's like an army, right? So if I ask you guys to fall in line, some guys will play, some guys will not fall in line. But if you are in the Chinese army, then there is no way, because the penalty is too high. You'll be shot probably. I hope so. No one is recording. Smriti, stop recording, please. All right, so it's sort of falling in line. Okay, it's rigidifying some local regions, and that is why there is too much of order, and that is why there is reduced fluidity.
Okay, sir. And so what causes the difference in the height or in the thickness?
Yeah, that's a good question. So it has been seen that in this lipid rafts region, there are lipids, I think, which are called as sphingomyelin. And I did not have it in my Did I have it? Let me check. No, I think there are sphingomyelin kind of lipids. They look different than the regular lipids. Okay, and that causes the thickness.
Okay. So springomyelin or sphingomyelin?
Sphingomyelin, sphingo.
Okay, sphingo. Okay. Okay.
Sphingomyelin. Yes, yes.
Okay. Thank
Not
you.
spring. Yeah. Anything else?
Sir.
Yeah.
Hello, sir. Am I audible?
Yes, yes, you're audible.
Sir, that linkage thing that you were explaining, the CACs and all, were you explaining it for the how we attach these fluorescent proteins to the lipids?
No, no, no. It was about how naturally occurring proteins are sort of farnesylated. Okay.
I sort of lost track over there at that point. Can you please somewhat re-explain in brief?
Why don't you ask some of your colleagues? They can explain you better.
Sir, not the process. Not the process, but the aim of that explanation.
No, because I was asked that how does it happen? So that's what I was explaining that. So can you see my slide here?
I see the lipid draft slide.
Yeah. No. Oh, okay. You see the lipid drafts.
Yes, this slide. The
Yeah. So what I was saying
vitamins.
that so there was a question that how does it happen, right? In terms of the reaction, in terms of the chemical functionality, how does it happen? So what I was saying that so you have phosphate groups. So this OH is phosphorylated, and if the
Sir,
OH
that I understood.
Then what is the question?
What was the reason behind you that explaining this thing? I am asking actually that.
How does it happen? How does the farnesyl group linked to the CAX motif?
Okay, now I got it. Thank you, sir.
Anything else?
So these, sir, only the membrane-associated isoprenes can undergo such reactions, or the vitamins also can undergo such reactions?
Although they look similar, I am not aware of any vitamins to undergo this kind of reaction. I'm not aware.
No, I mean in general, not this CAX motif, but because you've included vitamins in this slide,
Yeah.
so I thought that both of them can be used for protein modifications, which would cause the proteins to shuttle between the membrane and the
Yes,
cytosol.
yes, I understand your question. I am not aware. I'm not aware. I mean, ignore, don't worry that, since they're in proximity, they have relation between them. But I am not aware of vitamins getting, vitamins are sort of tethered onto proteins to get it attached. Although I will not be very surprised if you come up with a with a literature reference which says that they have found this.
Sir, and initially, while after discussing that CAX motif, or before that, you were discussing about how the vitamins can be used in to transport proteins from the membrane to the cytosol, and cytosol to the membrane, and then you shifted to the other to the associated.
No, this one you're talking about.
No, at the same slide, the initial slide, like A and B. Yeah. So initially, we were talking about vitamins, which can be used to shuttle the proteins, and then you
No,
talked about
vitamins are not used to shuttle the proteins.
I didn't get those 2 or 3 sentences which you said.
No, what I said that I'm sorry if I said that, but I really doubt. Vitamins are not used to shuttle proteins. Proteins can help vitamins shuttle.
So how is that possible? Yeah, that is where I got confused.
That's how. Are you seeing my slide?
Yes.
Yeah. So this is how. right? For example, it can be even clarified by this. For example, this is a cholesterol transport protein, and cholesterol molecule, well, vitamins do not exactly, but ergosterol and testosterone, these are not vitamins, but these are steroids. So they are, so if they are required from one part to the other part of the protein of the cell, so they can be transported by specific protein transporters. The only reason is, they're very hydrophobic and they'll not be soluble in cytosol. They will rather prefer to stick to the membrane. And if you are to detach them from the membrane, like as I said that, so detergents are used to solubilize proteins from the membrane environment to the external environment. So likewise, proteins are present, specialized proteins. They can, sort of snatch the vitamins from one membrane and take it to, their site of action.
So you mean to say that proteins can be used to transport vitamins, and the membrane-associated isoprenes can be used to transport the proteins?
Yes.
Okay.
Anything else? Okay. If there are no more questions, then I will post these slides. I will ask Sarah and Smriti to post these slides. You can see these slides and read further. Are there any more questions? I see a hand raised. Now I'm seeing this window, Teams window. Okay, it's gone. All right, great. So, okay, so Wednesday we'll start with the glycans. If you can read about glycans and all. All right, guys, take care. Bye.